System of respiration begins from nasal cavity, nasopharynx, oropharynx, larynx, trachea, different bronchi and ends in lung. Till larynx it is upper respiratory tract and distally it is lower. Coarse contaminations in the air are trapped by the hair in the nasal cavity and dust particles are trapped by the mucosa. Proximal part of the respiratory tract is conductive and distal part where exchange of the gases happen, that is respiratory part. Pocket-like extension of nasal cavity into the skull bones form paranasal air sinuses. They help in humidification of air. Vocal cord of the larynx are actually watchdog of respiratory tract. They prevent entry of liquids and solids into the lower respiratory tract. Phonation is acquired biological function. Trachea and bronchi have cartilaginous structures in their wall to prevent the collapse. In the upper part of this tree, it also has some secretory glands and pseudostratified ciliated columnar epithelium to trap the dust particles. Lower down, in the bronchioles, there are no cartilages and no glands. Trachea divides into primary bronchus to each lung. On the right side, primary bronchus divides into 3 lobar bronchus. They are also called secondary bronchus. On the left side, only 2. The segmental bronchus are the one which aerate the bronchopulmonary segments. Segmental bronchus are shown with different colors in this plastic cast. Without cartilages and glands, the bronchiole divide in several hierarchy to form an anatomical conductive airway where it has some pockets like openings. Here we see these are alveolar pockets which are opening on the bronchiole. This is respiratory bronchiole. Just proximal to it, it is terminal bronchiole. Cross section through trachea shows hyaline cartilage in the wall. Towards the lumen, it is pseudostratified ciliated columnar epithelium with goblet cells. Lamina propria shows some seromucous glands. Diagrammatically, the same epithelium, glands in lamina propria, hyaline cartilage with perichondrium and cell nests. This is scanning electron microscopic view of cilia of the epithelium and goblet cells. The dust particles which are trapped in the mucus which is covering on these cilia is continuously beaten towards pharynx. That is called mucociliary rejection current. When the section is through the lung, we see different conducting parts and alveolar exchange part. This is bronchus having cartilage in the wall. We can see here there are cartilaginous pieces, seromucous glands and pseudostratified ciliated columnar epithelium with few smooth muscles. This is bronchus. And few bronchioles which are devoid of cartilages. Bronchiole has no cartilage but smooth muscles are there. Epithelium is changing to simple cuboidal epithelium and there are no glands. After several hierarchy, terminal bronchiole and then respiratory bronchiole, alveolar ducts, alveolar sacs and alveoli. This is how this is respiratory bronchiole, alveolar duct, alveolar sac and openings into particular alveoli. And there are some spores which are connecting two different alveoli to maintain the pressure in all alveoli equally. This is bronchus because there are cartilages and glands in the wall. This is bronchiole because there are no cartilages and no glands but more smooth muscles and some connective tissue. Epithelium gradually changes from bronchus to bronchiole and terminal bronchiole into pseudostratified ciliated columnar epithelium. Slowly it becomes columnar epithelium, then short columnar epithelium, then cuboidal epithelium, short cuboidal, and finally towards the exchange area it becomes squamous. In the conducting part, cartilage goes on diminishing, muscle goes on increasing, and elasticity goes on increasing till the distal alveoli where it is more elastic. Even glands become lesser and distally there are no glands in bronchiole and in alveoli there are no glands. Scanning electron microscopic view of bronchiole, alveolar duct and alveoli. In histological sections it is terminal bronchiole, respiratory bronchiole as there are pockets of alveoli opening, alveolar duct, alveolar sac and alveoli. Alveoli are richly covered outside by capillaries and elastic fibers. These are different alveoli with interalveolar septae having capillaries lining endothelium, and these are alveolar squamous epithelium type 1 pneumocytes, and these are type 2 pneumocytes which are cuboidal which secrete surfactant, and there are alveolar macrophages, and these are the outer limits for margins for squamous cells lining the alveoli, and these are the pores of Kohn which are interalveolar communicating pores through which macrophages can pass and even equalization of the air pressure happens. This is very high magnification of diffusion barrier. You can see here erythrocyte, and this is endothelium of the capillary. This is basement membrane, which is common for alveolar epithelium, which is here, type 1 pneumocyte, and the nucleus of the type 1 pneumocyte is here.